Hello students, welcome to the PD Life Science Academy for the Happy Learning. NEET एक्झामचे प्रिपरेशन जे विद्यार्थी करत आहेत त्यांच्यासाठी बेसिक कन्सेप्ट लर्निंग आणि लिंकिंग ऑफ कन्सेप्ट हे जेवढे गरजेचे आहे तेवढेच NCERT बुकमधील चॅप्टरचे कंटेंट रीड करणे आवश्यक आहे. हे करत असताना फक्त एक रीडिंग करून चालणार नाही, मल्टिपल टाइम्स रिव्हिजन होणे गरजेचे आहे. यात टाळाटाळ करणे चांगले नव्हे. आपण सेट केलेले टार्गेट अचीव करण्यासाठी PD लाईफ सायन्स अकॅडमीने सर्व स्टुडंटसाठी जे नीट एक्झामचे प्रिपरेशन करीत आहेत त्यांच्यासाठी 21 डे रीडिंग चॅलेंजची सुरुवात केली आहे. रोज एका चॅप्टरचे रीडिंग आपण कंप्लीट करणार आहोत. हे चॅलेंज ज्या विद्यार्थ्यांनी ॲक्सेप्ट केले आहे त्यांनी त्यांचे डेली टार्गेट अचीव केल्यावर कॉमेंट करून सांगायचे आहे. सो लेट्स गेट रेडी फॉर टेकिंग रीडिंग चॅलेंज. ऑल द व्हेरी बेस्ट. थँक यू. 21 डे बायोलॉजी रीडिंग चॅलेंज डे 19. Reading challenge chapter number 19 from NCERT biology textbook of 11th standard. Name of chapter is a circulatory product and their elimination. Animal accumulate the ammonia, urea, uric acid, carbon dioxide, water and the ions like the sodium, potassium, chloride, phosphate, sulfate, etc. either by the metabolic activity or by means like the excess ingestion. In this chapter, we will learn the mechanism of the elimination of these substances with special emphasis on the common nitrogenous waste like the ammonia, urea, uric acid are the major forms of the nitrogenous waste excreted by the animals, and the ammonia is the most toxic form and requires the large amount of the water for its elimination, whereas the uric acid, being the least toxic, can be removed with the minimum loss of the water. The process of the excreting ammonia is the ammonotelism, and many bony fishes, aquatic amphibians and the aquatic insect are ammonotelic in nature. The ammonia, it is readily soluble and is generally excreted by the diffusion across the body surface and through the gill surface in the fishes as ammonia ion. The kidney do not play any significant role in its removal. The terrestrial adaptation necessitates the production of the lesser toxic nitrogenous waste like the urea and the uric acid for the conservation of the water. The mammal, many terrestrial, amphibian and the marine fishes mainly excrete the urea and are called as a ureotelic animals. Ammonia produced by the metabolism is converts into the urea into the liver of these animals and released into the blood, which is a filter and excreted out of the kidney. Some amount of the urea may be retained in the kidney matrix of the some of these animals to maintain the desired osmolarity. Reptiles, birds and the snails and the insect excrete the nitrogenous waste as the uric acid in the form of the pellet or the paste with the minimum loss of the water and are called as a uricotelic animals. Survey of animal kingdom present a variety of the excretory structures. In most of the invertebrates, these structures are the simple tubular forms, whereas the vertebrates have the complex tubular organs called as a kidney, and some of these structures are mentioned here. The protonephridia or a flame cells are the excretory structures in the platyhelminthes or flatworms, for example planaria, rotifers, some annelids and the cephalochordates. The amphioxus. The protonephridia are the primarily concerned with the ionic and the fluid volume regulates, that is the osmoregulation. The nephridia are the tubular excretory structure of the earthworm and the another annelids. The nephridia help to remove the nitrogenous waste and maintain the fluid and the ionic balance. The Malpighian tubule are the excretory structure of most of the insect including the cockroach. The Malpighian tubule helps in the removal of the nitrogenous waste and the osmoregulation. Antennal glands and the green glands perform the excretory function in the crustacean like the prawns. The 19.1 is a human excretory system. In human, the excretory system consists of the pair of the kidney, one pair of the urethra and the urinary bladder. The kidney are the reddish brown bean-shaped structures situated between the levels of the last thoracic and the third luminar vertebra, close to the dorsal inner wall of the abdominal cavity. Each kidney of the adult human measure the 10 to 12 centimeter in the length, the 5 to 7 centimeter in width, and 2 to 3 centimeter in kidney width, and the average weight of the 120 to 170 grams. Towards the center of the inner concave surface of the kidney is the notch called as the ileum, through which the ureter or a blood vessels and the nerves enter. The inner to the ileum, the broad funnel shaped space called as a renal pelvis, and projection called as a calyx. The outer layer of the kidney is the tough capsule. Inside the kidney there are the two zones, an outer cortex and the inner is a medulla. Medulla is divided into the few conical masses, that is the medullary pyramids, and projected into the calyxes. Single is the calyx. The cortex extends in between the medullary pyramids as a renal column called as a column of the Bertini. Each kidney has the nearly 1 million complex tubular structures called as a nephrons, which are the functional unit. Each nephron has the two parts, the glomerulus and the renal tubules. The glomerulus is the tuft of the capillaries formed by the afferent arterioles, the fin branch of the renal arteries, and the blood from the glomerulus is called as away by and the efferent arterioles. The renal tubule begins with the double wall cup-like structure called as a Bowman's capsule, which encloses the glomerulus. Glomerulus along the Bowman's capsule is called as a Malpighian body or a renal capsule. The tubules continue further to form the highly coiled networks, that is the proximal convoluted tubule, that is the PCT. The hairpin shaped Helen's loop is the next part of the tubules, which has the descending and an ascending limb. The ascending limb continue as the another highly coiled tubular region called as a distal convoluted tubule, that is the DCT. The DCT is a many nephron opens into the straight tubule called as a collecting duct, many of which converts and opens into the renal pelvis through the medullary pyramids in the calyx. The Malpighian corpuscle, the PCT and the DCT of the nephron are situated the cortical region of the kidney, whereas the loop of the Henle dips into the medulla. The majority of the nephron, the loop of the Henle is too short and extend only very little into the medulla. Such a nephron are called as a cortical nephron, and in the sum of the nephrons, the loop of the Henle is a very long run deep into the medulla. These nephron are called as a juxtamedullary nephron. Each efferent arteriole emerging from the glomerulus form a fine capillary network around the renal tubule called as a peritubular capillaries. A minute vessel of the network run parallel to the Henle's loop form a U-shaped vasa recta. The vasa recta is absent in a highly reduced in a cortical nephron. 19.2 is a urine formation. Urine formation involves the three main processes, namely the glomerular filtration, reabsorption and the secretion, that take place in the different parts of the nephron. The first step in the urine formation is the filtration of the blood, which carried out by the glomerulus and called as a glomerular filtration. On the average, the 1100 to the 1200 ml of the blood is a filter by the kidney per minute, which constitute roughly the 1/5 of the blood pumped out of each ventricle of the heart is a minute. The glomerular capillaries blood pressure causes the filtration of the blood through the three layer, that is the endothelium of the glomerular blood vessels and the epithelium of the Bowman's capsule and the basement membrane between these two layers. The epithelium cell of the Bowman's capsule, called as the podocytes, are arranged in the intricate manner so as to have some minute spaces called as a filtration slit or a slit pores. The blood is filtered so finely through these membrane and that almost all constitute of the plasma except the protein passes on to the lumen of the Bowman's capsule. Therefore, it is considered as a process of the ultrafiltration. The amount of the filtrate formed by the kidney per minute is called as a glomerular filtration rate, that is the GFR. The GFR is a healthy individual is approximately 125 ml per minute, that is the 180 liters per day. The kidney have built in a mechanism of the regulation of the glomerular filtrate rate. One such efficient mechanism is carried out by the juxtaglomerular apparatus, that is the JGA. Juxtaglomerular apparatus is a special sensitive region formed by the cellular modification in the distal convoluted tubule and the afferent arteriole at the location of their contact. A fall in the GFR can activate the the juxtaglomerular cell of the release the renin, which can stimulate the glomerular blood flow and thereby the GFR back to the normal. The comparison of the volume of the filtrate formed per day, that is the 180 liters per day, with that of the urine release, the suggestion, that is the 1.5 liters, suggests that the nearly 99% of the filtrate has to be reabsorbed by the renal tubes. This process called as a reabsorption. The tubular epithelial cell is a different segment in the nephron perform this either by the active or a passive mechanism. For example, the substances like the glucose, amino acid, sodium, etc. in a filtrate are absorbed actively, whereas the nitrogen wastes are absorbed by the passive transport. Reabsorption of the water also occurs passively in the initial segment of the nephron. During the urine formation, the tubular cells secrete the substances like the H+ and K+ and the ammonia into the filtrate. The tubular secretion is also an important step in the urine formation, as it helps in the maintenance of the ionic and the acidic base balance of the body fluids. 19.3 is a function of the tubules. The proximal convoluted tubule, that is the PCT. The PCT is a line by the simple cuboidal brush border epithelium, which increase the surface area of the reabsorption. Nearly all the essential nutrients are the 70 to 80% of the electrolytes and the water are reabsorbed by this segment. The PCT also help in the maintain the pH and the ionic balance of the body fluid by the selective selection of the hydrogen ion, ammonia and the potassium ion into the filtrate, and by the absorption of the HCO3- from it. The Henle's loop, the reabsorption is a minimum in its ascending limb. However, this region play a significant role in the maintenance of the high osmolarity of the medullary intestinal fluid. The descending limb of the loop of the Henle is permeable to the water but almost impermeable to the electrolytes. This concentrates the filtrate as it moves down. The ascending limb of the impermeable to the water but allows the transport of the electrolytes actively or passively. Therefore, as the concentrated filtrate passes upward, it gets diluted due to the passage of the electrolytes to the medullary fluids. The distal convoluted tubule, that is the DCT, conditional reabsorption of the sodium and the water take place in this segment. The DCT, that is the distal convoluted tubule, also capable of the reabsorption of the carbonate and the selective secretion of the hydrogen and the potassium ion and the ammonia to maintain the pH and the sodium potassium balance in the blood. Now the collecting duct, this long duct extends from the cortex of the kidney to the inner parts of the medulla. The large amount of the water could be reabsorbed from the region of the region to produce a concentrated urine, and this segment allows the passage of the small amount of the urea into the medullary interstitium to keep up the osmolarity. It also plays a role in the maintenance of the pH and the ionic balance of the blood by selective secretion of the H plus and the K plus ion. 19.4 is a mechanism of the concentration of the filtrate. The mammal have the ability to produce the concentrated urine. The Henle loop and the vasa recta plays a significant role in this. The flow of the filtrate in the two limbs of the Henle loop is in the opposite direction and thus form the counter current. The flow of the blood through the two limbs of the vasa recta is also in the counter currents pattern. The proximity between the Henle loop and the vasa recta as well as the counter current in them help in the maintaining and increase the osmolarity towards the inner medullary interstitium, that is the from the 300 milli osmomoles per liter. In the cortex, about the 1200 moles osmomoles per liter in the inner medulla. This gradient mainly caused by the NaCl and the urea. NaCl is a transport by the ascending limbs of the Henle loop, which is the exchange with the descending limb of the vasa recta. NaCl is a returning to the interstitium by the ascending portion of the vasa recta. Similarly, the small amount of the urea enters the thin segment of the ascending limb and the Henle loops, which is the transported back to the interstitium by the collecting tubules. The above describe the transport of the substances facilitated by the spatial arrangement of the Henle's loop and the vasa recta, also is called as the counter current mechanism. The mechanism helps to maintain the concentration gradient in the medullary interstitium. The presence of such a interstitial gradient help in and the easy passage of the water from the collecting tubule, thereby the concentrating the filtrate, that is the urine. The human kidney has produced the urine nearly the 4 times concentration than the initial filtrate form. 19.5 is a regulation of the kidney function. The functioning of the kidney is efficiently monitored and regulated by the hormonal backward mechanism involving the hypothalamus, JGA, and to certain extent the heart. The osmoreceptor in the body is activated by the changes in the body blood volume, the body fluid volume, and the ionic concentration. An excessive loss of the fluid from the body can activate these receptors, which stimulate the hypothalamus to release the antidiuretic hormone, that is the ADH, and the vasopressin from the neurohypophysis. The ADH facilitates the water reabsorption from the latter parts of the tubule, thereby preventing the diuresis. An increase in the body fluid volume can switch off the osmoreceptor and suppress the ADH release to complete the feedback. The ADH can also affect the kidney function by its constrictory effect on the blood vessels. This causes an increase in the blood vessels. An increase in the blood pressure can increase the glomerular blood flow and thereby the GFR, glomerular filtration regulator. The JGA plays a complex regulatory role, falls in a glomerular blood flow or a glomerular blood pressure, or a GFR can activate the G cells to release the renin, which converts the angiotensinogen in the blood to the angiotensin 1 and further to the angiotensin 2. Angiotensin 2 being powerful vasoconstrictor increase the glomerular blood pressure and thereby the GFR. Angiotensin 2 also activate the adrenal cortex to release the aldosterons. The aldosterone cause the reabsorption of the sodium and the water from the distal part of the tubule, and this also leads to an increase in the blood pressure and the GFR. The complex mechanism is generally known as the renin angiotensin mechanism. An increase in the blood flow to the atria to the heart can cause the release of the atrial natriuretic factor, that is the ANF. ANF can cause the vasodilation or a dilation of the blood vessels, thereby the decrease a blood pressure. ANF mechanism therefore act as the check on the renin angiotensin mechanism. 19.6 is the micturition. The urine formed by the nephron is ultimately carried to the urinary bladder, where it is stored till the voluntary signal is given by the central nervous system, that is the CNS. The signal is initiated by the stretching of the urinary bladder as it gets filled with the urine. In response, the stretch receptor on the wall of the bladder and the signal to the CNS, and the CNS passes to the motor message to initiate the contraction of the smooth muscle of the bladder and the simultaneous relaxation of the urethra sphincters, causing the release of the urine. The process of the release of the urine is called the micturition, and the neural mechanism causes it is called as a micturition reflex. An adult human excrete on an average 1 to 1.5 liter of urine per day. The urine forms as a light yellow color watery fluid which is a slightly acidic, that is the pH is a 6, and has the characteristic odor. On an average 25 to 30 grams of the urea is excreted out per day. Various conditions can affect the characteristic of the urine. Analysis of the urine helps in a clinical diagnosis of the many metabolic disorders as well as the malfunctioning of the kidney. For example, the pressure of the glucose and the glycosuria and the ketone bodies or the ketonuria in the urine are the inactivated of the diabetes mellitus. 19.7 is the role of the other organ of the excretion. Other than the kidney, the lungs, livers and the skin also help in the elimination of the excretory waste. Our lungs remove the large amount of the CO2, approximately 200 ml per minute, and also significant quantities of the water every day. Liver, the largest gland in our body, secrete the bile containing the substances like the bilirubin, cholesterol, degrades the steroid hormones, the vitamins and the drugs. Most of these substances ultimately passes out along with the digestive wastage. The sweat and the sebaceous gland in the skin can eliminate a certain substances through their secretion. The sweat produced by the sweat gland and watery fluid containing the NaCl, small amount of the urea, lactic acid, etc. Though the primary function of the sweat is to facilitate the cooling effect on the body surface, it also help in the removal of some of the waste mentioned above. The sebaceous gland eliminate the certain substances like the steroids, hydrocarbons and the waxes through the sebum, and this secretion provide a protective oily covering for the skin. Do you know that the small amount of the nitrogenous waste could be eliminated through the saliva too? The 19.8 is the disorders of the excretory system. The malfunctioning of the kidney can lead to accumulation of the urea in the blood and condition called as a uremia, and which is highly harmful and may lead to the kidney failure. In such a patient, the urea can be removed by the process called as a hemodialysis. During the process of the hemodialysis, the blood drain from the convenient arteries is pumped into the dialyzing unit called as the artificial kidney. The blood drain from the convenient arteries is pumped into the dialyzing unit after the adding and the anticoagulant like the heparin. The unit contains the coiled cellophane tube Surrounded by the fluid, it is the dialysing fluid having a same comparison as that of the plasma except the nitrogen waste. The porous cellophane membrane of the tube allows the passage of the molecule based on the concentration gradient, as the nitrogen waste are the absent in the dialysing fluid. These substances freely move out and thereby cleaning the blood. The cleaning blood is a pump back to the body through the veins and affects the adding antiheparin to it. This method is a boon for the thousands of the uremic patients all over the world. The kidney transplantation is the ultimate method in the correction of the acute renal failure, that is the kidney failure. A functioning kidney is used to transplant from the donor, probably a closer relative, to the minimize its chance of the rejection by the immune system of the host. The modern clinical procedure have the increased the success rate of the such a complicated technique. The renal calculi, the stone or a insoluble mass of the crystalline salt, that is the oxalate etc., form within the kidney. Glomerular nephritis, that is the inflammation of the glomeruli of the kidney. Now the summary of the excretory product and their elimination. Many nitrogen containing substances, iron, CO2, water etc., that accumulate in the body have to eliminate. The nature of the nitrogen waste form their excretion vary among the animals, mainly depending on the habitat, that is the availability of the water. Ammonia, urea, uric acid and the major nitrogen waste is excreted. The nephridia, protonephridia, nephridia, Malpighian tubule, green glands and the kidney are the common excretory organ in the animals. They not only eliminate the nitrogen waste but also help in the maintenance of the ionic and the acidic base balance of the body fluid. In human, the excretory system consists of the one pair of the kidney, a pair of the ureters, a urinary bladder and the urethra. Each kidney has over a million tubular structure called as a nephron. The nephron is a functional unit of the kidney and has the two portions, glomerulus and the renal tubules. The glomerulus is the tough and the capillaries formed from the different afferent fine branches of the renal arteries. The renal tubules start with the tubular wall and the Bowman's capsule and is further differentiated into the proximal convoluted tubule, that is the PCT, hence the Henle loop, that is the HL, and the distal convoluted tubule, that is the DCT. The DCT, the many nephrons join to the common collecting duct, many of which ultimately opens into the renal pelvis through the medullary pyramids, and the Bowman's capsule encloses the glomerulus to form the Malpighian or a renal capsule. The urine formation involves the three main processes, that is the filtration, reabsorption and the secretion. The filtration is the non-selective process performed by the glomerulus using the glomerulus capillary blood pressure. About the 1200 ml of the blood is a filter by the glomerulus per minute to form 125 ml of the filtrate in a Bowman's capsule per minute, that is the GFR. JGA, the specialized portion of the nephron, plays a significant role in a regulation of the GFR. Nearly 99% of the reabsorption of the filtrate take place through the different parts of the nephron. PCT is the major site of the reabsorption and the selective secretion. HL primarily help in the maintaining the osmolar gradient and the 300 milli osmomole per liter to the 1200 milli osmomoles per liter within the kidney interstitium. The DCT and the collecting duct allowing the extensive reabsorption of the water and certain electrolytes, which helps in the osmoregulation. H+, K+ and ammonia could be secreted into the filtrate by the tubules to maintain the ionic balance and the pH of the body fluid. A countercurrent mechanism operates between the two limbs of the loop of Henle and those of the vasa recta and capillary parallel to the Henle's loop. The filtrate gets concentrated as it moves down the descending limb but is diluted by the ascending limb. The electrolyte and the urea are retained in the interstitium by this arrangement. The DCT and the collecting duct concentration, the filtrate about the 4 times, that is from the 300 milliosmomoles per liter to the 100 and 200 milliosmomoles per liter. An excellent mechanism of the conservation of the water. Urine is stored in the urinary bladder till the voluntary signal from the CNS carried out its release through the urethra. That is the micturition. The skin, lung and the liver also assist in the excretion. Thank you so much.